So, good afternoon, everyone. I'm Saranjeev Rangaraj. I'm from Department of Biochemistry, University of Hyderabad. I'm a PhD scholar and a PMR of scholar as well. To do my teaching assistantship, I've associated with this NPTEL teaching assistantship, and this is the Doctorate section 7, and we are in the week 7 as well. And the course that we have associated so far is overview and then integration of cellular metabolism. We'll get into the subject today. As usual, we'll see recap on week 7 concepts and some practice questions we'll do. And if someone is there, then we can do the doubt clearing session as well. And the topic we have started with carbohydrates in the past 6 weeks. Then we have arrived at the lipid metabolism and lipoprotein metabolism. Now we are starting protein. So we are going to start how the digestion of proteins will take place and what are the different proteases available in the life, right? And finally, we'll see the some absorption of the protein from gastrointestinal tract. And if you see the protein molecule, it can be taken up from anywhere, like through meat or leafy vegetables or any kind of thing that we are eating. It does contain protein molecules. So the dietary protein is maybe it's from the plant or it can from the animal origin as well. So for example, we usually take 50 to 100 gram per day. So the essential amino acids should only taken from the diet. Essential amino acids are the amino acids which cannot be produced in our body. So those amino acids should be taken from the diet itself. And there are other amino acids called non-essential amino acids. It can be synthesized in the body or it can be derived from the diet. And the endogenous protein from the digestive tract also it should contain about 30 to 100 grams per day. And the 70 kg man has an average protein turnover rate of 400 grams per day. Because this digestion or the protein turnover in the body itself, the amino acid pool can be generated from anywhere. It can be digestion of the enzymes that is present in the body, or the killing of the protein molecules, or degradation of the protein molecules through various mechanisms like proteolysis, autophagy, or in the case of the cell death, it can be happen in any way. The final form that we are going to get it is as an amino acid. So the digestion and then the absorption of the protein is very efficient. Hence, very little protein, that is 5 to 10 gram per day, is lost through feces. And then the body amino acid pool is always in a dynamic state. In an adult, the rate of synthesis of the protein balances the rate of degradation. So the nitrogen balance is maintained. So these are the proteolytic enzymes, those are present in the body. If you see, there is no proteolytic enzyme in saliva. So that's why the gastrointestinal tract is very, very important for the uptake of the amino acid pool. And in the stomach, if you start, there are pepsin and then the renin will be there to do the proteolytic activity. And in pancreas, we will be having the four different set of amino enzymes. For example, trypsin, chymotrypsin, elastase, and then the carboxypeptidase A and B. And finally, the intestine, that is aminopeptidase, dipeptidase, and then the tripeptidase. And most of the proteolytic enzymes are synthesized in zymogen form. The reason is the production of the enzymes are in somewhere else, and then the action of the enzymes will be in the somewhere else. So this we have elaborately seen from the first class itself, like how or from the first week itself we have seen how where these enzymes are getting secreted or produced, then where it is getting secreted or it gets its active form, that we have seen already. And if you see the proteolytic enzymes, as in the example, the peptidases are two types. One is the endopeptidase and another one is exopeptidase. Endopeptidase is nothing but if this is the amino acid structure in the body, the endopeptidase cut the amino acid in between the chain. Whereas exopeptidase only targets either in the N terminal or in the C terminal of the peptide molecule. That we are going to see what are the things are going to happen. So endopeptidases, they upturn peptide bonds inside the protein molecule, forming the small peptide fragments. For example, pepsin, trypsin, chymotrypsin, and then the elastase. Whereas exopeptidase, which act only the terminal peptide bonds, and this group includes carboxypeptidase and then the aminopeptidase. So these are the proteases that we have seen, exopeptidase and as well as the endopeptidase. Endopeptidase will cut the amide bond in between the protein molecule, whereas aminopeptidases or the dipeptidyl peptidases or tripeptidyl peptidases will cleave the three amino acids in the uh, terminal end, and then the carboxypeptidase also can be can cleave the amino acids as one or two are the dipeptidases and tripeptidases. This is what the action that people have seen. And omega peptidases, we know that omega is the farthest molecule will be cleaved. And if you see in the stomach pH is very, very low, which is around 2 to 3, which is an acidic pH due to the secretion of the hydrochloric acid. There, the enzyme called pepsin is getting activated. So, due to the presence of the acidic pH, the pepsinogen form of pepsin, that is an inactive form, pepsinogen, will be converted to pepsin by this active, and that will be denature the protein. And as we know that pepsin is the endopeptidase and this is secreted by the chief cells of the stomach. And the activation that we have seen, this will be removal of the 44 amino acids will take place at the internal end. And this pepsin catalyzes the address of the bonds formed by the carboxyl group of phenylalanine, tyrosine, and tryptophan and methionine and converts the protein into the protease, proteoses, and then the peptones. And there is one more enzyme that is present in the stomach that is called renin, which is otherwise called as the chymosin. This usually active in the infants, not in the adults, and this helps in the curdling of milk. And this is absent in the adults, and then milk protein casein is converted to the paracasein and combined with calcium, and denature the protein is easily digested further by pepsin. And next, digestion in the small intestine takes place by the different enzymes. It is a mixture of the endopeptidases as well as the exopeptidases. And endopeptidases, for example, trypsin, chymotrypsin, elastases are called serine proteases. These serine proteases are having the serine amino acid in their active sites, so-called serine proteases in the terminology itself. And then finally, the carboxypeptidase, that is the exopeptidase. And if you see, the secretion of the pancreatic juice is stimulated by the peptide hormones, that is cholecystokinin and then the pancreozymin. And this secreted as a zymogen, and secretion of the alkaline bile and then pancreatic juice is optimum around pH 8. And from there, this whatever the juice that we have seen, pancreatic juice, that is really important form that will activate the enzyme called enterokinase, and that will get activated by the removal of the hexapeptide from the N-terminal line. And this activate the trypsin. Okay, and there are two kind of activation people have seen. One is the autocatalysis of the trypsin because trypsin itself can cleave and then it can get activated. And finally, trypsin catalyzes the hydrolysis of the bonds formed with the carboxyl groups of arginine as well as the lysine. And if there is any kind of issue in the trypsin activation that leads to the acute pancreatitis, that is the leads to the premature activation of the trypsinogen inside the pancreas itself. That will result in the autodigestion of the pancreatic cells. And this is what that we have seen trypsinogen by the removal of some amino acid which is called hexapeptide amino acid from the internal end that leads to the activation of the trypsin. And this trypsin, it goes and activates a certain other enzymes called chymotrypsinogen to chymotrypsin transfer, proelastase to elastase, and procarboxypeptidase to carboxypeptidase, prolipase to and then lipase. And the first enzyme that gets activated, or whatever the mechanism that we are going to see, that is chymotrypsin. And then this gets activated by the trypsin molecule. This is very, very important in terms of any kind of enzymes. First, the chymotrypsinogen is around the 245 amino acid peptide, which has 5 intra disulfide bond, And whenever the trypsin gets activated, this trypsin will cleave the protein at the 15 position, that is where the arginine 15 is there. Then this will form the active enzyme called 5-trimotrypsin, and this again by the action of the cell digestion by the 5-trimotrypsin that will cleave the part in the case of that will leave the dipeptide, that is 147 and then 146, that is tyrosine, and then the leucine 13, and then the asparagine 148, and that leads to three different fragments, and that are linked by the two intra-chain disulfide bond. So the reason that I'm asking, focusing on this thing is people will ask so many questions in that the activated alpha chymotrypsin has five disulfide bond totally. In that, two are inter and three are intra disulfide bond, because this is really really important for the exam perspective. In the exam perspective, this is very very important thing. And next enzyme, that is elastase, the same enzyme that is trypsin activate the elastase, and it is also an endopeptidase, and then the zymogen form of elastase is proelastase, and it starts on the peptide bonds formed with amino acids like glycine, alanine, and then serine. And carboxypeptidases are the exopeptidases, and the procarboxypeptidase is the zymogen form of carboxypeptidase, and it is also gets activated by the trypsin. And this requires the enzyme, it's a metalloenzyme, and it requires the metal called zinc. And carboxypeptidase A cleaves the preferential, the aromatic, and then the branched chain amino acids, whereas carboxypeptidase B cleaves preferentially positively charged amino acids. And these are the table. This is the table of or the comprised version of the different proteases, and then their mode of action, where it goes and then how it is gets activated. So if you see pepsin acts on the aromatic amino acids and then the acidic amino acid, that is glutamic acid, and trypsin is the endopeptidase that acts on the basic amino acids, that is arginine and then lysine. And chymotrypsin acts on the aromatic amino acid, that is tryptophan, phenylalanine, tyrosine, and then methionine. And elastase acts on neutral amino acids, that is glycine, serine, and then the alanine. And carboxypeptidase usually works on the aromatic amino acid and then the branched chain amino acid, that is tyrosine, phenylalanine, tryptophan, valine, leucine, and then isoleucine. And carboxypeptidase works only on the positively charged amino acid, that is called basic. And inside the small intestine, there are other enzymes also present, that is called aminopeptidases, as well as the dipeptidase and then the tripeptidase. And finally, the overall digestion of amino acids or protein molecule, if this is a protein molecule, finally it will give one single unit of amino acid. So that will get absorbed through different cycles in the body. So the absorbance of amino acids mainly takes place in the small intestine of the body, and it is Na plus, that is sodium dependent active transport system, where ATP is involved. And sodium amino acid is getting co-transport with the amino acids. There are five different carrier systems are there in the body. For example, first is the neutral amino acids, second one is the basic amino acids, and then the amino acids, and then glycine, and acidic amino acids, and finally beta amino acids, example beta alanine. And one important cycle that takes place in the inside the membrane of the small intestine, or in the, yes, small intestine, that is gamma glutamyl cycle, otherwise it's called as a Meister cycle. That is really really important for the absorbance of the amino acids inside the body. So the glutathione is a molecule, it is a tripeptide, it's made up of three different amino acids, that is glutamate, cysteine, and then glycine. It's otherwise called as a gamma glutamyl glycine. This is the only like the exception of the peptide bond making in the amino acid dipeptide, because where usually alpha carbon is involved in the peptide bond, but in the case of the glutathione, that is involved in the gamma carbon. So gamma glutamyl cystinyl glycine, and it reacts with any amino acid. For example, here if you see one amino acid is in the outside the membrane, it's coming inside the membrane, and that binds to the glutathione. Then it forms the glutamyl amino acids. Then the cystinyl glycine will be removed. Okay, from here. Then this glutamyl amino acid, by the action of the gamma glutamyl cyclotransferase, it transfers the amino acid inside the cell. Then that forms the oxoproline. This oxoproline, in the presence of the 5-oxoprolinase, again it will form the glutamate, where one ATP molecule is involved, and again from the glutamate, by the action of gamma glutamyl cysteine synthetase, again it will form the glutamyl cysteine. Then this glutamyl cysteine, in the presence of the glutathione synthetase, then one molecule of ATP again it's utilized. Then the glutathione will be formed. So it is a very active process and then the energy consuming process as well. If we see the clinical importance in this case, the deficiency of the enzyme 5-oxoprolinase, which is here in this pathway, leads to the oxoprolinuria, because the excretion of the pyroglutamic aciduria will takes place. The allergy to the certain foods proteins, that is milk and fish, absorption of the partially digested proteins will take place. That leads to so much of the allergic reactions of the body. And there are the defects in the intestinal amino acid transport systems are seen in the inborn errors of the metabolism, such as the heart disease, immunoglycinuria, and then the cystinuria. And then the certain partial gastrectomy, pancreatitis, carcinoma of the pancreas, and cystic fibrosis may affect the digestion and absorption of the proteins. And the protein losing enteropathy, there is an excessive loss of proteins through the gastrointestinal tract as well. And next, if the amino acids are taken inside the cell, then what is the fate of it? Like if it is there, then it has to go for the other pathways, or it should be helpful for the other clinically or the physically important molecule. So that is what we are going to see, how this amino acids are going to be really helpful in the body. So that can happen through deamination or the transamination reaction, or decarboxylation reaction, or in the case of transdeamination reaction. So, if you see the overall the protein catabolism, the protein lysis of the dietary proteins on the stomach, and then the small intestine releases the free amino acids in the bloodstream. That's what we have seen so far. Then it can also comes from the proteolysis of the proteins that move through the endocytic pathway, takes place in the lysosome of the all cells. Uncontrolled proteolysis, that is ubiquitin tagged proteins also, that gets into the proteasomal degradation mechanism, that also finally form the amino acid pool. And if you see the structure of the amino acid, this is one amino group will be there, and then the one carboxylic group will be there, and then the side chain will be there in the system. And how this is getting denatured, or the catalysis will catabolism will happen. Either the amino group will be removed, or in the case of carboxylic group will be removed. That we are going to see in the case of the deamination, transamination, and then the decarboxylation reaction. All of the reaction, all of the opposite reaction usually takes place in the liver. So the deamination of the amino acids usually takes place. That is the elimination of the amino group from the amino acid with ammonia formation. And four types of deamination takes place. That is the oxidative, the most important for higher animals, and then the reduction, and the hydrolytic, and then the intramolecular deamination reaction. And these are the examples people have given. For example, in the case of the reductive deamination, there is a NH3 will be released. In the case of the histidase, then a urocanoic acid will be formed. And then second one is because of the addition of H plus ions, and then the hydrolytic deamination because of the desulfurase. One such example, L-cysteine, by the action of the desulfurase will give the amino acids. Then again the H2O will be added. Then the NH3 will be removed. Then finally the pyruvic acid will form. And finally intramolecular deamination, that leads to the one such example is serine, by the action of the removal of H2O. that will give the amino acids. Then the amino acids again H2O will be formed. Then the pyruvic acids will be formed. These are the different examples of reductive, hydrolytic, and then the intramolecular deamination. And one such deamination that we have already talked about is the oxidative deamination. That is really, really important in the case of deamination reaction in the body. This L-glutamate dehydrogenase it's usually present in the mitochondria of the liver. Okay, and what it does usually, it uses both NAD plus and then the NADP plus as a cofactor. And here, glutamate, by the action of glutamate dehydrogenase, by forming the chief base as an intermediate, then it forms the alpha ketoglutarate by removing one ammonia molecule. Okay, and L-glutamate dehydrogenase is a metalloenzyme which requires the zinc as a uh, metalloenzyme, and then it has accept the coenzymes such as NADP, DP plus or NAD plus, and it consists of the six identical subunits in the structure. And then allosteric regulation of this thing is GTP as well as the ATP is an allosteric inhibitors of GDH, whereas GDP and then the ADPs are allosteric activators. And there are other hormones like steroid and then the thyroid hormones inhibit the glutamate dehydrogenase and low level of energy that is decrease the ATP and increase the catabolism of amino acid and alpha ketoglutarate as a substrate for PCA cycle. And metabolic sequence of this GDH that is usually the reversible in action. And so it takes place in the both anabolic as well as the catabolic reaction. And catabolism channels nitrogen from the glutamate to urea, whereas the anabolism takes place amination of the alpha ketoglutarate by the ammonia to glutamate. And there are different transformation that the next very next reaction is we have seen the oxidative deamination. Now we are going to see how this transformation reaction takes place. The transformation reaction is the most common that can take place by the alanine aminotransferase or as the aspartate aminotransferase. An alanine aminotransferase, what it does, it takes place alanine plus alpha ketoglutarate, and then it will finally give transfers the amino group from the alanine to the alpha ketoglutarate. Then it forms glutamate, and then finally the pyruvate will be released. The same way, aspartate plus alpha ketoglutarate by the action of aspartate aminotransferase, otherwise called as AST, then it forms the glutamate and it releases the oxaloacetate. So usually the aminotransferase funnel the alpha amino groups from the variety of amino acids to the alpha glutarate and then with glutamate formation, and this glutamate can be deaminated with ammonia release from the previous reaction that we have seen. And this transaminase reaction, alanine transaminase or aspartate transaminase reactions are sorry, transferase. These reactions are need the prosthetic group called pyridoxal phosphate, that is PLP. So this takes place in the two different reactions. Usually, what happens if A molecule is there and then the B molecule is combined with this, then the AB intermediate will be formed. Then finally CD will come. But here it is quite different mechanism takes place. What will happen? This aspartate molecule, which is an amino acid, that gives the amino group to the cofactor that is PLP, that is pyroxyl phosphate, that forms the pyroxamine phosphate. And from here, this pyroxamine phosphate takes place this amino acid or this amino group that will be given to alpha ketoglutarate to form the glutamate molecule. Even though the participating molecules are aspartate as well as alpha ketoglutarate, there is a ping pong mechanism that takes place where this PLP molecule gets aminated by NH2, then it transfers the amino group to the other reactant that is called alpha ketoglutarate, then it forms glutamate. Then from our aspartate, directly oxaloacetate will be formed. So that's why aspartate plus alpha ketoglutarate forms oxaloacetate and then the glutamate. And if you see the clinical significance of this kind of reactions, the aspartate aminotransferase are induced by the glucoparticles, which favors the gluconeogenesis. And AST is increased in the myocardial infarction as well as ALT in the liver disease. And all of the amino acids except the lysine, threonine, and protein, and then hydroxyproline participate in this kind of transformation reaction usually. And if you see the biological significance of this transformation, this is the first step of the catabolism of amino acid because it is removing the ammonia group. And then the first step, ammonia is removed, and the carbon skeleton of the amino acid enters the catabolic pathway. And synthesis of the non-essential amino acids also takes place because by means of the transformation, all non-essential amino acids can be synthesized by the body from the keto acids available from the other sources. For example, pyruvate can be transformated to the synthesis of alanine. Similarly, oxaloacetate produces aspartic acid, and alpha-ketoglutarate is transformated to form the glutamic acid. And those amino acids which cannot be synthesized in this manner are therefore essential, they should be made available in the food. And these are the interconversion of the amino acids. If you see, these are the essential amino acids, these are the non-essential amino acids, synthesis from the essential amino acids, and these are the non-essential amino acids which can be synthesized in the body itself. And finally, the trans-deamination reaction. So this it's a combination of two things. First, the transformation reaction will take place, then followed by the oxidative deamination. So due to the transformation reaction, it forms the glutamate that we have seen, and from there it can go to the liver. Then the deamination will take place because glutamate, by the action of glutamate dehydrogenase, which can use both NAD plus or NADH. DP or NADH as a cofactor, then it forms the alpha beta glutarate. There also ammonia will be released. So the transdeamination reaction, the transformation takes place in the cytoplasm of the cells, whereas the amino group is transported to the liver as glutamic acid, which is finally oxidatively deaminated in the mitochondria of hepatocytes. So only the liver mitochondria contain the glutamate dehydrogenase. That is the reason it's very, very important. So the two components of the reactions are physically far away, but physiologically they are coupled in transdeamination. And this is the same thing, like how this ammonia is formed, the same we have given it in this different form. And finally, the decarboxylation. Decarboxylation is the name itself, is the removal of the carboxyl group from the system. So if any amino acid is there, then it will remove the carboxyl group. So that leads to carbon dioxide removal, then the amine will be formed. So usually amines have the high physiological activity, hormones, neurotransmitters, etc. So the enzyme involved here is decarboxylase. Here again the coenzyme which is the pyridoxal phosphate, that is called PLP, pyridoxal phosphate. And one such example or two examples that we have given here, one is glutamate by the action of the decarboxylation reaction that form the GABA. GABA is the gamma aminobutyric acid, and it's a mediator of the nervous system and which is inhibitory in nature actually, inhibitory neurotransmitter. Whereas the histamine is formed from the histone, like yeah, not histone, it's formed by histidine amino acid. So by the decarboxylation of histidine amino acid, it forms the histamine, and this is a mediator of the inflammation and allergic reactions in the body. And this is the overall fate of the carbon skeleton. And it either form the pyruvate or it can form the acetyl CoA that we are going to see in the next week. Which are the glucogenic or which are the ketogenic or which are the both. The glucogenic are amino acids which will give the intermediates of the glycolysis or gluconeogenesis, whereas ketogenic amino acids usually give the acetyl CoA. Then how it will proceed the reaction that we are going to see. And finally, the metabolism of the ammonia that we are seeing. The ammonia can be formed in any case, like we have seen in so many cases, the ammonia will be formed. The sources of ammonia cells in the different cases, and then different varieties are there. For example, the first such example that we have seen in the last few slides is the transformation reaction. That finally, this will give the ammonia as a byproduct. That is first. And second, the reductive deamination or the hydrolytic deamination or the intermolecular deamination also, finally the end product will be the respective product as well as the ammonia group. And finally, from the glutamine also, the kidneys from the ammonia form ammonia from the glutamine by the actions of the renal glutaminase. So this glutaminase, what it will use by the action of the H2O molecule, like by the involvement of the H2O molecule, it will remove the NH3. So that also form glutamate in this case. So most of the ammonia are excreted into the urine as maintaining the NH4 plus and the body's acid base balance will be maintained. And third one is it can from the bacterial action because in the intestine the bacteria contains the urease enzyme, so that can also release the ammonia molecule. And from amines also it can be synthesized because the amines that we have seen in the last, like the histamine or the gamma, whatever it is, there also it can be synthesized as a byproduct, this ammonia, and from purines and fibrins also. Whereas this ADA will be there, adenine deaminase, those things. So these ADA, those will form from the AMP, it will form the IMP, and then there also release of ammonia will be favored. So in these cases, ammonia are released. And finally, one more thing is L-amino oxidase, and this requires FMN as a cofactor. There also an alpha keto acid will be formed, and again the ammonia will be released. So these are the very little of importance, and then the other minor pathways are histidine to the urocanoic acid and NH3 by the histidase, and asparagine to the aspartate, and serine and threonine to pyruvate, and finally cysteine undergoes the deamination and transsulfuration to form the pyruvate by the enzyme desulfidase. And even though the ammonia are formed here and there, and then it has to be, it's really toxic to the cell, and then it has to be metabolized. So how it will be metabolized? Usually, the metabolism of this ammonia takes place in the liver, but the enzymes responsible for this ammonia detoxification is not present in anywhere else. So what happens in the muscle tissue? It has to be converted as alanine cycle. So how it will be done that we are going to see. So that is otherwise called as the Cargill cycle. What will happen in the system? This ammonia, whatever it is there, free ammonia, that forms the glutamate. Then the glutamate, the by the action of the pyruvate, then it transfers the alanine aminotransferase activity will be there, then it will be synthesized as alanine. This alanine passes through the blood and it reaches the liver. There, the urea cycle will take place again. And finally, ammonia transported in the form of the glutamate itself, usually in the brain. What will happen? This L-glutamate, by the action of the glutamine synthetase, this form the gamma glutamine phosphate, and again by the action of the glutamine synthetase, it forms the L-glutamine. This glutamine, what will happen? Here only the ammonia addition will be there. This glutamine can travel in the system. So this by the action of the glutaminase in the liver mitochondria, then it will again form as a glutamate. And what are the ammonia is released from the system? Again, it will go to the urea cycle. And in the blood system, if you see the circulating glutamine levels, it's found in plasma. It's higher than the other amino acids. And if you see this glutamine synthetase, which is really, really important because that helps in the detoxification in the brain. So the regulation of the glutamine synthetase is also really, really important. What will happen? This glutamate can be regulated by glutamine synthetase can be regulated by any number of the feedback mechanism that has mentioned in this figure. So once the glutamine is formed, then it can form again different molecules like CTP, histidine, glucosamine 6 phosphate, carbonyl phosphate, tryptophan, and AMP, and everything that we will see it in the later classes. How these things are really, really important in the real case. And once the ammonia is there, ammonia can be formed in the different forms. It can form the alpha ketoglutarate. It can bind to the alpha ketoglutarate and then finally form. Yeah. So it can binds to the alpha ketoglutarate or combine with that, then finally it forms the glutamate. Then it finally forms the other amino acids. Or else this again bind to the glutamate, then it's formed glutamate, and again it can go to the nucleotide synthesis, cytidine nucleotides, amino sugars, and tryptophan, and then they studied. And it can combine with aspartate and then form the asparagine, or else it can combine with CO2 and in the presence of ATP, then it forms the carbonyl phosphate. Then it can form the arginine or pyrimidines or the urea. So this is the overall mechanism that we have seen so far. And here one small topic that we are going to discuss, that is why ammonia is really toxic to the brain. If you see, ammonia usually reacts with glutamate, and then the alpha ketoglutarate to form glutamate. Then the depleted levels of alpha ketoglutarate will take place in the brain system. Then the tricarboxylic acid pathway will not take place. Due to that, the energy level will be very low if the more ammonia is produced in the brain. Then the glutamate, whatever is formed here, that can use to form the glutamine formation. If the glutamate is not there, then the GABA will not be produced, which means the gamma aminobutyric acid. If these two things are not here, then what will happen? The depletion of the inhibitory neurotransmitters will take place. And whenever the glutamine exchange happens in the body, it happens through tryptophan exchange. Because if one glutamine is outgoing, then the tryptophan will come inside. This tryptophan is the precursor for the hormone called serotonin. Sorry, the neurotransmitter called serotonin. This serotonin is the excitatory neurotransmitter. Then the more excitatory neurotransmitter leads to the more activation of the system. And if you see in the brain cells such as the astrocytes, that contains most of the total brain glutamine synthesis, and that leads for the glutamine synthesis. And the well neurons contain virtually all brain glutaminase. That's why excess glutamine leads to the osmotic imbalance in astrocytes, and water intake swells of the astrocytes, leading to the cerebral edema. And raised intracranial tension can lead to coma as well. And if you see the ammonia induced activation of the NMDA, that is N-methyl D-aspartate receptors, increases the nitric oxide formation, which in turn reduces the activity of glutamine synthase, and then thus reducing the elimination of the ammonia in the brain. And this is the overview of thing. And if you see whatever the final thing, whatever the protein that we have taken, that again forms the ammonia, then ammonia will come to the urea cycle. That's what we are going to see now. What is the urea cycle? How it will be removed? The removal of amino group also will take place. That will see how the nitrogen group is removed due to the transamination reaction or deamination reaction. Whatever the reaction, it can form the ammonia group will be removed. This is the removal of amino group 1. And usually that can be transported through the secretion system or in the blood system through the cell. So the transport of the glutamate releases amino group as ammonia in the liver. Finally, it forms because that we have seen in the last slide. So the amino groups from the many of the alpha amino acids are collected in the liver and the form of the amino group and of the glutamate molecules. So the glutamate dehydrogen is what it will do. Then it is in the mammalian liver has the unusual capacity to use either the NAD plus and then the NADP plus as a cofactor. The nitrogen carriers usually in the body or in ammonia carriers in such case. Glutamate that transfers the one amino group within the cells, this one, and then the another one is glutamine that transfers the two amino group between cells and releases the amino group in the liver, and alanine transfers the amino group from the tissue, that is muscle, and then into the liver system. And this is the overall thing. And L-glutamate, by the action of the glutamine synthetase, because of the synthetase, the ATP molecule will be spent here. Then the gamma glutamine phosphate will be there, and then again the glutamine synthetase is formed, and it will transverse the L-glutamine. And this glutamines move between the cells. Then it will go into the liver. Then finally it enters the urea cycle, and again the L-glutamate will be formed here. And this is the overall thing, that the entry of the nitrogen into the mitochondria, how it will happen. The glutamate is formed here in the different ways. Then it gets into the mitochondria of the cell. This glutamate, again by the action of the glutamate dehydrogenase, again it will form the ammonia. So by various sources, the glutamate will be formed. Then they prepare the nitrogen to enter the urea cycle is this glutamate. It will by the action of the glutamate dehydrogenase forming. Then the first step will be takes place in the urea cycle where that bicarbonate molecule by the action of the two ATP molecule molecules expenditure, then finally the carbonyl phosphate will be synthesis. That is what we are going to see as a urea cycle as usual. That it is otherwise called as Krebs encelate urea cycle or ornithine cycle as well. The site of action or the site usually takes place in the liver. And there are subcellular organelle will be there. The first few reactions will take place in the mitochondria. Then it comes to the cytoplasm. And then the first metabolic cycle to be elucidated in the system is that is urea cycle, and it converts the toxic ammonia into the harmless urea. So this is the step one. How this is happens by the action of the carbonyl phosphate synthetase. Two molecules of ATP is used in this case. This ammonia, carbon dioxide, and then the H2O. Actually, it's this carbonyl phosphate synthetase one is involved in the urea cycle. Then it forms a carbonyl phosphate. And from this carbonyl phosphate, by the addition of this ornithine, because it forms in the as a citrulline, so it forms carbonyl phosphate plus ornithine, then it forms a citrulline by the action of ornithine transcarbamylase. And again, in the citrulline is inside the cytoplasm. Then again, the addition of the aspartate will takes place. This aspartate addition will leads to organosuccinate synthesis by the action of organosuccinate synthase. Here, one molecule of H2O will be removed as well as one ATP molecule is involved. And here, if you see, one AMP is uh, formed, where usually ADP is formed, but here is AMP formation. And finally, this organosuccinate by the release of fumarate, and then the organosuccinate lyase will be there. One fumarate molecule will be released, and then the arginine will be formed. And again, arginine by the action of araginase enzyme, then it forms urea. And one here also one addition of H2O molecule, water molecule takes place, and one of the ammonia molecule is from the released thing, another ammonia molecule is from the aspartic acid molecule. And this is the overall urea cycle. You can stop here and then check what are the different molecules are involved, how it takes place, and everything so far. And this is the overall review. If you see carbonyl phosphate that is involved in the formation in the mitochondria is a prerequisite for the urea cycle, where carbon dioxide plus ATP and then ammonia molecule form the carbonyl phosphate, and where ATP molecules involved because it's a synthetase. And hence, citrulline formation from the carbonyl phosphate and then ornithine done by the ornithine transcarbamylase. And aspartate provides the additional nitrogen to form organosuccinate in the cytosol. That organosuccinate synthetase is involved here. Again, arginine and then by the addition of the fumarate, by the action of the organosuccinate lyase, this arganosuccinate will be formed or lysed into arginate and then the fumarate. And finally, the hydrolysis of the arginate takes place to urea and then ornithine, that is by the action of arginase. And overall chemical balance, if you see, ammonia and then carbon dioxide and three ATP molecules are involved here. Then urea is formed, two ADP and AMP, and then two inorganic phosphate and one PPA will be formed. And this is the overall reaction that we have seen so far. And if you see the energetics in the urea cycle, two ATPs are formed, and in the first reaction, another ATP is converted to AMP and then PPA, which is equal to two ATPs. So the urea cycle consists of four high energy phosphate bonds. However, fumarate formed in the fourth step may be converted to the malate cycle. Malate, when oxidate to the oxidized state, produces a 1N80H, which is equal to 2.5 ATP. So now net energy expenditure is only 1.5 high energy phosphate here. And what are the formed here? This fumarate that I used to tell here. Fumarate again it will go to the shunt pathway, small shunt pathway, then it form the malate and then the oxalic acid, and again it will enter the TCA cycle. Okay. So the significance of the urea cycle, if you see, if it disposes the two waste products, that is usually the ammonia as well as the bicarbonate, and it forms the semi-essential amino acid that is arginine. And because of the reason, because the arginine is really important, for the cycle. A ornithine is regenerated following synthesis. There was role in the cell growth and then proliferation, and non-essential amino acid synthesis also needed because ornithine is a precursor of protein molecule. And if you see the disorders of the urea cycle, the genetic defect is usually has been described in all enzymes of the urea cycle, results in ammonia intoxication. The one such reaction that if you see, it's very rare and extremely rare. If you see 1 in 30,000 live births, and autosomal recessive disorder is in one such cases, all but except OTC defect because OTC defect is X-linked trait. A defect in the reaction 1 and 2 it leads to the accumulation of the ammonia directly, whereas defect in the later forms are accumulation of the intermediates. And this is the overall thing. If the carbonyl phosphate synthetase 1 deficiency is there, and which is autosomal recessive, then the arginine levels are reduced and then citrulline levels are reduced in the plasma. And urine organic acid, normal urine uric acid will take place. Whereas ornithine transcarbamylase deficiency is there, that is OAT, which is X-linked actually, that leads to arginine and then citrulline reduction. And arginosuccinic acid synthetase deficiency, whereas arginine levels are upregulated and then the citrulline is also upregulated. Other functions have been given here. You can stop the video and then check it that what are the reactions that takes place. And first one is hyperammonemia, that is type 1. The defective enzyme here is CPS1, and then the products accumulated are ammonia. So here we can see severe hyperammonemia, mental retardation, and developmental delay also takes place. Whereas variant of the hyperammonia type 1, where N-acetyl glutamate synthetase, that is synthesis the gene defective, there also ammonia products accumulated. Then neonatal hyperammonemia, fatal it's actually, and then N-carbamide L-glutamate activates the CPS1. And hyperammonemia type 2, which is OTC X-linked, that also the product accumulated is ammonia, which leads to the uric acid urea, challenging the CP to pyrimidine synthesis. And next one is hyperammonemia type 1, and there also CPS1. Sorry, these are the same things actually. You can go back to this videos and then check it what are these things. I think I have that much only for the class. And if you go back to the questions now, which cycle is important for amino acid absorption, which means which is very used in that. If you go back to the first slide and then check it, that is Meister cycle, okay, and which is really active process. So that is gamma glutamate cycle, otherwise called as Meister cycle. Not TCA cycle, not glycolysis, not urea cycle. These things are involved in the carbohydrate metabolism. And next one is which enzyme cleaves most of the proteolytic enzyme. If you go back and then check the slides, the enzyme really important for the enzyme is that is trypsinogen. What happens? Trypsinogen by the action of enteropeptidase, it forms the trypsin. Then the trypsin cleaves the other enzymes like chymotrypsinogen to chymotrypsin, proelastase to elastase, procarboxypeptidase to carboxypeptidase, and lipase to lipase. So that is the reason trypsin is really important in the case of proteolytic cleavage. So trypsin is really important. So the most cleaves the most of the proteolytic enzymes are it's trypsin, not pepsin, not chymotrypsin, and then the non elastase. And next, which cycle removes ammonia from the body? Yeah. So if you see alanine cycle takes ammonia from the tissue to the cell, but it will not remove the ammonia from the body. So glycolysis, you know that it's a catabolic pathway of the glucose where it forms a pyruvate. So it can also cannot remove. PCA cycle, you know that it's a tricarboxylic acid cycle involved in the carbohydrate metabolism. That is also not correct option for this question. So another urea cycle where urea by the action of carbon dioxide, ammonia, and then the water molecule from the carbonyl phosphate in the mitochondria. Then from the carbonyl phosphate, Again, the other reactions like ornithine, arginosuccinate, arginine, and then these things will be formed in the urea cycle. And finally, which enzyme uses both NADPH and NADH as cofactors? That is not carbonyl phosphosynthase synthase 2 that is involved in the primitive metabolism. And finally, the glutaminase. Glutaminase also it's not there, which is a kidney specific enzyme that is also not used both. And then arginine also will not use both. Only one enzyme is left that is glutamate dehydrogenase. That is the reaction. It is otherwise called as G D H. So I think that is what we have today. If any doubts are there, then we can go back and let's see. So if I go back once again, the overall, I think once we take the any protein molecules inside the body, that can get into the saliva, but the saliva cannot does not contain any kind of protease enzyme, so it cannot be digested in the mouth. So it will get into the stomach. The stomach will have the pepsin and then renin. Then the pepsin, what it will do, it will get activated by the action of the HCl. Then it will form the active form of the pepsin from the pepsinogen. Then it can catalyze the hydrolysis of the certain peptide bonds like phenylalanine, tyrosine, and tryptophan methionine. Then it forms the peptones. Then the renin will be there, renin, but it's really active in the infants, but not in the adults. It's not present in adults actually. Then in the small intestine, there are variety of enzymes will be there. One such enzyme is the trypsin, which will get activated by the endokinase by the removal of the hexapeptide. This specific for the arginine and then the lysine, and it goes and binds to the different goes and activates different kind of proteases. Like for example, chymotrypsinogen, proelastase, procarboxypeptidase, and prolipase. And if you see the one overall view that how these different peptidases will go and then digest a different set of amino acids in the body. And we have seen the absorptions of the amino acid as well. There are different carriers there in the cell to take up the amino acids in the small intestine. One such example that we have seen that cycle is Meister cycle. So Meister cycle is the gamma glutamyl cycle that takes the glutamyl amino acids and then the glutamate and glutamyl cysteine, and finally the glutathione will be formed. Here, amino acids will take place by the glutathione, then it will remove the cysteine and glycine, and from the cysteine and glycine, glycine and cysteine will be formed, whereas gamma glutamyl amino acids will be formed. Again, by the action of the cyclotransferase, oxoproline will be formed, then the amino acid will be removed. So this is what the overall cycle. This is very very important cycle for the amino acid absorption. And clinical importance also we have seen what will happen. And there are certain amino acid degradation of the catabolism should take place where it should remove the amino acids where it can do by the two set of reaction. One is the amino uh, transamination of the removal of the amino group or the removal of the carboxyl group. So we have seen the deamination reaction where the oxidative deamination is really really important. We have seen the glutamate deamination is there, which is really really important for the glutamate synthesis in the body, and we have seen the how it is has a different set of identical subunits. And finally, we have seen that what are the different kind of transformation reaction takes place by ALT and AST, and these enzymes are requested the pyridoxal phosphate. Then how this ping pong mechanism takes place in case of the transformation enzymes, and their same clinical significance that we have seen. And finally, we have seen that how this trans-deamination reaction takes place. That is a two-step reaction, but the one takes place in the different segment, and the other takes place in the different segment. So that we have seen in the this thing. And finally, the decarboxylation of the amino acids we have seen that leads to the certain amine groups that is called gamma, otherwise GABA, otherwise the histamine. How that relates to the neurotransmitter as well as the mediator of the inflammation that we have seen. And finally, once the ammonia is formed from the different sources, whereas in the removal of the transdeamination or the deamination, reductive deamination, or from the glutamine, or from the bacterial sources, from amines or the purines and pyrimidics, and from some minor pathways that we have seen how the ammonia will get formed. Then finally, we have seen that how this ammonia, formed ammonia, is transferred to the liver for the detoxification, either through the alanine cycle or through the glutamine formation that we have seen. And how the glutamine synthesis is really, really important. What are the inhibitors of glutamine synthesis that we have seen? And once the ammonia is formed, how it will be utilized also we have seen. And finally, how why ammonia is really really toxic to the brain that we have seen. And finally, how this ammonia formed in the body, which will transfer to the uh, liver, and how it will again form the ammonia from the glutamate and form the carbonyl phosphate. Then how it is forming the citrulline by the addition of the ornithine, then genosuccinate, then the arginine, then finally the urea formation that we have seen it in the overall reaction. And finally, the which is the enzyme defect in the body. And finally, this urea cycle uses the 1.5 energy phosphate to function normally. And finally, we have seen the significance of the urea cycle, then the disorders of the urea cycle with certain questions that we have seen. So, this is what the overall thing for the week 7. We will see in the next class how these things are really, really important, and then how these things are going to help in the upcoming amino acid synthesis pathway. So, that is called anabolic pathway. So, we'll see you next week.